Hi guys, welcome to Mido's Bio. This channel is dedicated to sharing information in an easy to understand way for free for biology competitions, especially the US Biology Olympiad. Today's video is about biotechnology. We will cover 21 techniques in good detail at a level that covers USABO Opens and Semifinals. Most of the information here is from Albert's Molecular Biology of the Cell, and other resources used, along with pictures, have all been linked under the corresponding technique. So here's how this video is organized. There are 21 techniques in 8 categories. The categories here are proteins, DNA, sequencing DNA, blotting and labeling techniques associated with blotting, and then looking at protein production, which is like ribosomes and transcription factors, and then molecular interactions, so how to measure how close two molecules are, and other technologies that didn't really fit in any one of those. Okay, so each technique will have a column that explains its purpose and has all the sources, and then there's a column with kind of the information, the methods, the explanations, and sometimes there are like vocabulary terms which will be bolded. And then there's a category here that is pictures. So these will help with kind of visualizing and aiding the explanation. And then lastly, there is a column for kind of how to read results and any notes on the pictures if it's applicable to the technique being explained. So let's get started. So first up is protein purification. The purpose of it is to isolate pure samples of proteins. So there's 2 steps. So first, you'll need to use fractionation, which is basically like blitzing the cells in a food processor. And this splits the big components of the cells apart without kind of damaging them internally. And you'll need to separate what you get out of that more, and you can use centrifugation or sedimentation to do that. So a centrifuge is basically this machine where you can put a tube of stuff in, like a suspension of cells or some blood, and have it spin it around really fast. And what this does is create an artificial gravitational field within the tubes that is much greater than like what it is on Earth. And this makes it so what would usually settle out eventually sinks to the bottom a lot faster. And the more and the faster you spin your cells, the smaller the pieces are going to be. So you can separate like different components of the cells at different levels. And yeah, so for sedimentation, you can do this with a gradient in a solution, and how fast the particles settle or where they like end up lying in the solution can separate them. So ideally what you would get out of this entire step is kind of a mixture of your desired protein and some other similar particles. So to further separate your solution, you could use column chromatography. And so what you use like in column chromatography is this column, and it's like open at this end here to drip out into like tubes. So this is what's like what you want dripping out. And then this is where you put like all of your solution you got from the first step. And in the middle there are these like beads that kind of filter out what you don't want or what you want. And so basically you're just going to end up with what you want here or what you want here. So there's 4 types of like chromatography columns. So there's an ion exchange column, and there's kind of like 2 names that it can have. So an anion exchange column is basically if what you're trying to pull out of the solution is an anion or negatively charged. And then a cation exchange column is the opposite. And basically for these, they'll like elute the column, which is basically wash everything that's not bound. So yeah. So in this case, like if it's an anion exchange column, then you'll have cations like bound to the beads. So the anions passing through will be bound to those beads, and then everything else gets washed off with like a salt solution or something. And then a second type of chromatography column is like an hydrophobic chromatography column. And here, like the beads are attached to like molecules with hydrophobic side chains. So what this does is basically it like attracts like in biology. So all the hydrophobic things in your solution are going to be attracted to the beads, and then everything else can be washed off. So that purifies those things. And then third is gel filtration, which is basically based on size. So it's a little bit counterintuitive how this works because the bigger molecules actually move faster through the beads. You'd think that they move slower because there's like more that can get in their way, I guess. But what happens is if you look over here, so the larger molecules, they can't get into the beads, so all they have to do is move around the beads. But smaller molecules, they actually can get like stuck in this polymer bead, so it takes them longer to get out of the solution. So yeah, and here's a picture showing that. And then affinity chromatography, it's basically you have a substrate covalently attached to your bead, and then essentially you kind of use that to pull what you want out of the solution, and then everything else ends up in like a little test tube. So yeah, that's protein purification. So next up we have SDS-PAGE. And the purpose of SDS-PAGE is to separate proteins by their size, and it's also the first step of a Western blot, which we'll talk about later. So the procedure of SDS-PAGE is pretty similar to DNA gel electrophoresis, but how you prepare the protein is kind of different. So we'll talk about DNA gel electrophoresis later. But in SDS-PAGE, what you have to do is you have to add a SDS and a reducing agent. So what SDS does here is it denatures your protein and it takes out any non-covalent interactions, and it removes the secondary, tertiary, and quaternary structures. So, and then another big thing it does is it gives your proteins like an overall negative charge that's like uniform. So later they'll be able to be attracted to like something positive. And then we have your reducing agent. So the most common one is beta mercaptoethanol. And what it does is it breaks disulfide bonds, if there are any, in your protein, and it turns it linear. So the combination of SDS and a reducing agent will essentially return your protein into a chain. And this makes it like easier for it to move in the gel and for you to see that like truly the distances represent the actual size, like the actual number of amino acids in your protein, and not just like how it's shaped and stuff like that. So your gel that you're going to be using here, what it's made out of is acrylamide, and you would add a crosslinker to that. So essentially you're making a mesh, and then that's called polyacrylamide. And changing the concentration can help you like kind of customize your gel for different sizes of proteins. So more like a larger concentration of polyacrylamide helps you with smaller proteins, because if you think about it like a sieve, if your mesh on a sieve is really big, like the holes are really big, then you can't really separate like small things that well because everything just falls through. But if your mesh is really small, then like you can separate, between the small and the very small very easily. So that's kind of like the principle that's going on here. And in your setup, you're going to have like your gel and your prepared proteins in here under like some buffer that's going to keep it at like a certain pH. And you're going to have it so the positive electrode is at the bottom and the negative electrodes at the top. So because we used SDS, all our proteins are going to be negative, so they're going to be attracted to the positive electrode. And then we would call this an electromotive force that moves the protein once there's electricity running through. And so once your gel is done running, what you have to do is stain it with a dye because you can't really see it otherwise. And the most popular one is Kumasi blue. So then you can look at your results and analyze them. So like in SDS-PAGE, larger molecules are going to move slower and the smaller ones are going to move faster. So the smaller ones are going to be closer to your anode, which is your positive electrode. And the bigger ones are going to be closer to the starting line because it takes them longer to move. So what you can do with the results of an SDS-PAGE is you can create like a calibration curve where you basically kind of run a set of proteins that like you know how big they are, and then you see how far they go in your gel. And then you can kind of like plot those using special information on the axes. And then you can plot like your unknown. Like for example, you have a protein that you're trying to identify based on like its molecular weight. Then you could plot that on and then see where it lies on the line of best fit here. And that is SDS-PAGE. So next up is 2D gel electrophoresis. And the purpose of this is to further separate proteins by looking at their isoelectric point and their size at the same time. So there's 2 steps to this. So each of them is 1 dimension. So the first one is isoelectric focusing. So what you need to do here is first treat your protein with something that can make it soluble and denatures it and dissociates all the change but does not change the charge. So you can do this with an uncharged detergent, beta-mercaptoethanol, and urea, and because urea denatures proteins. So regarding the uncharged detergent point, so if you remember SDS from earlier, that's also a detergent, but it's charged because it gives your proteins a overall negative charge, so they'll run through the gel properly. However, isoelectric focusing kind of depends on a protein's like charge by itself, so you want kind of its own natural charge. So the next step is to separate the proteins in a pH gradient. And what you're trying to do here is you're trying to identify the isoelectric point. And that's basically the point, like the pH, when your protein has no net charge. And you can identify this because like in your tube, you're going to have like a pH gradient here. And you're going to have your proteins like somewhere in the middle. And then you're going to apply like an electric charge on both sides. Like here you can see this is positive, this is negative. And then it's going to go up and down based on its charge. But once it reaches like the pH that it's like where it has no charge, it's going to stop moving because it's not attracted by the electric field. So that's like a really clever way to kind of identify the isoelectric point. And there's going to be special buffers in this to kind of control the pH since it's such a like big part of this. And then that's isoelectric focusing. So the second step is electrophoresis, which is kind of like SDS-PAGE from earlier. But what you do is you take this tube you used to do IEF, and then you're going to turn it at a 90-degree angle, and then you're going to run it through your gel. Like, right, so you're going to do an SDS-PAGE here. And what happens is your proteins, they're already separated like in this dimension, and then they're going to be separated in this dimension now. So this lets you identify like proteins that might have the same size but not the same isoelectric point. Or they might have the same isoelectric point but not the same size, because it gives you these 2 data points kind of at the same time. So it's really useful for kind of really similar proteins. And that's it for 2D gel electrophoresis as well as proteins as a whole right now. So now we're going to look at some DNA techniques. So the first one we'll look at is DNA gel electrophoresis. And this is really similar to SDS-PAGE from earlier when we were talking about proteins. So the purposes of DNA gel electrophoresis can be to identify like similarities in sets of DNA based on like the length of restriction enzyme cut fragments. And what restriction enzymes are, they're basically these like molecular scissors that cut at specific DNA sequences. So these are going to be different sequences for different people. So unless you're identical twins. So the links they're going to cut the DNA into are going to be different. And this can be used for DNA fingerprinting, which we'll talk about next. And then DNA gel electrophoresis can also be used to determine the sequence of like a short strand of DNA. So first thing you do is you cut up pieces of DNA with restriction enzymes. And then the second thing you'll do is mix it with some loading dye. So I forgot to mention this earlier with SDS-PAGE, but loading dye is also used there. So the purpose of it isn't to dye like your DNA or protein. It's basically there so when you're running your gel, you can see like kind of the line of dye like moving down or moving across. And this is important because DNA and proteins, they're colorless, and you don't want to run them off the gel if you run your gel for too long. So this dye kind of shows you how things are progressing. And then your third step is to put like your mixture of loading dye and DNA into your wells and in your gel here. So your gel is covered in a buffer just like with SDS-PAGE. So you'll stick a pipette in and then you can inject it in one of these wells. And for your gel, there's different gels you can use for DNA gel electrophoresis. So if your strand of DNA is short, then you'll use polyacrylamide like you did for, SDS-PAGE. But if your DNA is long, then you will use agarose, which is like a polysaccharide that comes from seaweed, I believe. And then there's a type of DNA gel electrophoresis you can use for really long strands of DNA called pulse field gel electrophoresis. And basically, you manipulate the DNA into like turning around at different times, and it takes longer pieces to longer to turn around. So again, like with SDS-PAGE, a larger percentage of like agarose or polyacrylamide works better for smaller pieces. So you run this with electricity, and the setup here is different than that for SDS-PAGE. I'm not sure why, but all of the protein gel electrophoreses are like vertical, whereas like the DNA ones are kind of like in a tray, like flat on a table. But yeah, you'll have all of your wells by the the negative electrode, the cathode, and then it's moving towards the anode at the end. But you don't need to use like SDS or anything because DNA is already negatively charged because of like the phosphates and the backbone. So yeah, once you're done running your gel, what you'll do is you'll dye it with ethidium bromide, which is like a dye, and you view this under UV light, and then you'll be able to see it. So when you're analyzing your data, you can see that bands closer to the anode are shorter, just like with SDS-PAGE, because they're able to move like a longer distance. And you may also be able to reference like a DNA ladder for the length of fragments. So here is a DNA like ladder or a marker. So basically each of these pieces represent like, chunks of DNA that are like this long. So you can see based on like this ladder where your fragments lie and stuff, and that kind of shows you how long they are. So if you look at this one, it's like pretty much in line with the 600 BP mark, so you can estimate that this one's about 600 base pairs long. And another another like technique you can do, like we said earlier, you can use DNA gel electrophoresis to determine a sequence. And what you do for that is you chop the DNA up basically, and you put it with some chain terminating nucleotides. So chain terminating nucleotides, uh, basically, so your normal nucleotides you can kind of like keep on building on top of them, but chain terminating ones, it's like you put that one on and nothing will go behind it. So basically, you have like identical DNA in each of the um like in each of these wells, and you mix in some chain terminating nucleotides, so only added like chain terminating adenines here, chain terminating cytosines, chain terminating guanines, and then thymines in here. And then what it'll do is like basically randomly mix with like randomly be incorporated at one point. And then once you run this, like these are being sorted by like shorter to longer strands of DNA. And then so your sequence is actually like CAAGT, like that. So that's a pretty cool way to determine a sequence. And yeah, that's about it for DNA gel electrophoresis. Okay, so next up is DNA fingerprinting. And what DNA fingerprinting tries to do is it tries to determine someone's identity based on like kind of segments of DNA that vary in people. So people have like really similar DNA, but there's just like, a few segments that are different, right? And then those kind of make us who we are. So it kind of analyzes those portions of DNA to see like if you can identify certain people. And it's often used in like forensics to, I think, rule out suspects for a crime. So there's kind of two ways you can do it. So the first one's with southern blotting, which we'll talk about later. But southern blotting is basically using DNA gel electrophoresis results and then visualizing them in a certain way. And then what it does basically is it looks at RIFLIPs, which are restriction fragment length polymorphisms cut by restriction enzymes. So RIFLIPs are basically like areas between where like restriction enzymes will cut that are like different in different people other than like identical twins. So if the length of your fragments like cut by restriction enzymes are the same as like another person's, then it's likely that you could be related and stuff like that. And the other method you can use is if you use PCR to amplify DNA. And what you're looking at are VNTRs, which I think stands for variable number tandem repeats. And what those are basically like humans have like these repeats in their DNA, and then different people can have like different numbers of repeats, so it analyzes those. And so as a general rule, relatives should have similar DNA fingerprints. And this can be important in certain types of analyses. And yeah, this is kind of a picture just showing how it can be used in forensics. So if you see that this person's matches this person's, then they could be the same person. And that's about it for DNA fingerprinting. So next up, we're going to talk about molecular cloning. So the purpose of molecular cloning is to amplify DNA, and it also helps you have kind of like copies of a desired segment of DNA at hand. So molecular cloning is like not the same as kind of like cloning an entire organism. It basically just entails making a bunch of copies of DNA. And basically what you'll need to do is you'll design a plasmid. So a plasmid is this like little circle of DNA you can find in bacteria. and they're like separate from the big chromosome, and they're just kind of little pieces of DNA, the bacteria kind of picked up. So what you'll need to do when you're designing your own plasmid is you cut your plasmid with a restriction enzyme, and then it becomes linear. And then you insert like the DNA you want to be cloned, right? So you cut with restriction enzymes like here, and then you put your gene in, and then you use ligase, which is um it's a enzyme that can kind of like covalently seal these ends back together. So what you've basically done is cut it open, like put something in, and then like stitch the ends back up. And then what you do is you reintroduce your plasmids back into bacteria, and then these bacteria, you've like you need to make them permeable to DNA so they can take it in. And then the way you retrieve your DNA that you want copied is by using the same restriction enzyme. So you just kind of cut it out again. And then, so like there's two plasmids that are kind of special, I guess. They're like favored because they're easy to use, and that's because they're big and there's not a lot of them in cells. So that means there's less likely to be recombination with like sequences on other copies of these types of plasmids. So it keeps your information more safe. And something you can do with plasmids is you can make a DNA library. So when you do this, you basically do the same process as when you were designing your plasmid. You have like one piece of DNA per plasmid, and then you put one plasmid per bacterium, and then you can just store them in like little wells on a plate. And then since you have to use restriction enzymes, I included a little section about them. So their purpose is basically to protect prokaryotes from foreign DNA by like cutting it up. But biochemists have kind of found new uses for them. And what they do is they cleave phosphodiester bonds on double-stranded DNA at specific palindromic sequences. So palindromic sequences are pretty interesting. Basically, if you look at this one, this sequence, like that, is the same as this sequence this way. And I'll skip down to here. So this exhibits like twofold rotational symmetry. Basically, if you like rotate this twice, you get the same sequence. So some restriction enzymes are going to cut at sticky ends, which are really convenient for if you're inserting something. So if we look at these enzymes, so these are just three that I've included. They're very popular ones for examples. So if you look at this one, Echo R1, it cuts like this. So you see how like it has like an end that's sticking out, so that's called like a sticky end. And if you're inserting DNA, you can just kind of like, you can have the end of your DNA match up with this portion so it sticks in really well. And lastly, I just have like a little piece of vocab that's probably useful. So cDNA is what happens when you use reverse transcriptase on mRNA. So reverse transcriptase is, it basically turns RNA into DNA. So basically this is DNA, but it has no introns. So you've got a gene, and usually you would have to splice the introns out, right? But this is already done for making mRNA, and just turning it back into DNA, it has no introns anymore. So that's about it for molecular cloning. Okay, so next we have PCR. So PCR, it's I'd say that it's a pretty common name now to hear because of COVID tests. But the purpose is to amplify and make copies of DNA, and it does this really fast. So basically, you need to use a DNA polymerase, which kind of extends strands of DNA, and it's you get this from the bacteria Thermus aquaticus, which is really good at withstanding high temperatures. So because there's going to be lots of temperature changes in PCR, you really want that. So, and PCR, it makes lots of copies really fast because it doubles the amount of DNA with each cycle. So you start off, say, with like 1, and then you get 2, and then you get 4, and then so on. It's exponential growth, right? So if we look at this, basically there's like 3 stages at different temperatures where different things happen. So first, you're going to take your double-stranded DNA, and then you're going to denature it, and then that turns it into single-stranded DNA, right? And then you're going to put in some primers that attach here, and then they'll only attach at like a lower temperature, so you'll have to lower the temperature for that. And then you can go back up for a higher temperature to extend the chains. And you do this with that DNA polymerase we talked about earlier. So yeah. And basically you just keep on doing that. You separate these strands again. And then if you look at this picture, it kind of shows like how fast it goes. Like, start off with 1, and then you get 2, and then you get 4, and you get 8, and then you make a bunch by like 20 or 30 cycles, right? So there's like different applications of PCR. So qPCR, which is quantitative PCR, it measures the amount of DNA present in each cycle with like probes or fluorescent dyes. And RT-qPCR, which is basically qPCR but with RNA, it measures the expression of genes with mRNA. So it kind of shows in a cell, like this gene is being expressed a lot, this one's not really being expressed, right? So if you'll remember, we mentioned primers here. So there's actually kind of names for each of these primers in PCR. So the forward primer is the one that attaches to the template or antisense or minus strand. So it's this one, and basically it makes lots of copies of the forward, like the plus strand, right? And then the reverse primer is like the opposite, and both of these always go like 5 to 3. So when you're answering questions on semifinals or something, you need to pay attention to what's being amplified. So amplified is I feel like it's a very vague term. But how I've seen on a past question is basically the primer begins the sequence that's being amplified. So if we look at the forward primer here, it's identical to the plus strand, or at least the beginning of it. So therefore it's amplifying this strand right here because it's on the opposite strand. So probably if they ask you a question like that again, then that's what they want. Okay. And that's it for PCR and also kind of just general DNA techniques. Then we'll move on to sequencing DNA. Okay, so first up is Sanger sequencing. So this was kind of one of the first technologies developed to determine the sequence of DNA. And this process is pretty simple. So what you do is you amplify your DNA with PCR, like we talked about, and then you denature into single strands. And what you're going to do is you're going to add normal nucleotides, which are called DNTPs, and then fluorescently labeled DDNTPs. So these are chain terminating, like we talked about earlier with I think it was gel electrophoresis for DNA. So these basically you put them onto a strand and then nothing's gonna come after them. So and in this case they're gonna have these fluorescent labels on them. And then what you're gonna do is you're gonna run all of these sequences through a capillary gel electrophoresis, which is kind of like the gel electrophoresis we talked about earlier, but it's in a really thin tube. And then you're going to have a laser pointed at this tube, and then the laser is going to detect kind of the fluorescence from the chain terminating nucleotides. So in more detail, basically what happens is that you have these like single stranded like pieces of DNA, and then they're just going to like grow with normal nucleotides until it hits like a termination one that's attached to a dye, right? So DNTPs until a DDNTP is reached. And because there's so many copies of DNA, just by chance there's basically going to be a bunch that are like, every length. Like there's going to be one that's just one nucleotide long, one that's two nucleotides long, right? And then so when you send it through your capillary gel electrophoresis, the laser is going to read like each of those ending like termination nucleotides, right? And then with that you can build a sequence. And then what it looks like is these peaks, and then you can analyze each of those for nucleotides. So one peak is one nucleotide, and there's a color for each of them, and then you just go across like that to identify your sequence. So the next technique is shotgun sequencing. So this is also to determine the sequence of DNA. And what you do in shotgun sequencing is you randomly chop up the DNA, and then you sequence like all of those short pieces right here. And then what you do is you stitch them together in silico, which basically means on a computer. So basically you sequence all these strands, and then you get the sequences, and then you use a computer to find like kind of these overlaps that have like the same sequence, and then it puts them together. So now we'll talk about next generation sequencing, or NGS, and this is really just so cool. So the purpose of this is basically to determine the sequence of DNA, but to do it faster than the previous techniques. And it uses either fluorescence or like detection of pH changes to do so. So I'll talk about Illumina and ion torrent sequencing, which are actually both trademarked kind of techniques. So Illumina is basically like a bunch of Sanger sequencing, but at the same time. So all the nucleotides used here are labeled fluorescently with a color, and they also all have a block that prevents the chain from getting longer. So this is a normal nucleotide, and this is kind of like an Illumina nucleotide. It's got your block and it's got a dye. So what happens is you basically have a bunch of like DNA that's like stuck onto like a little tray. And then what you do is you basically wash like a bunch of nucleotides over it, a bunch of like your special ones, and then it's going to attach, and then the color you're going to see like that fluoresces is like the one that was just attached, right? And then what you're gonna do is you're gonna take a picture, and then you're gonna after washing away all the other nucleotides, and then you're gonna take a picture. And then what you're going to do is you're going to remove the block, and you're going to remove the dye. And then you're going to wash it again with nucleotides and just keep on going. So each time you can only add one nucleotide, and there's only one dye like on one sequence because you're taking it off every time. And then so you have a photo after each cycle, and then a computer kind of like looks at those photos and it's like, oh, green, then yellow, then red. That means like this sequence. So it's kind of a really efficient way to do like your normal sequencing, but like all at the same time. It's super cool. And then next is ion torrent. So what you do here is you have like an array with these little wells. So here's one well, and then in each of them you have like a little bead that's coated in like identical copies of DNA, and then you have a semiconductor chip underneath. So basically what happens is you wash each nucleotide over. So you wash like, a bunch of adenines over, a bunch of, thymines over. And then when like the reaction to add a nucleotide, it releases a H+, it releases a proton. And this causes a change in pH just in like each one of these little wells that this chip can detect. So through detecting these changes in pH and like kind of associating with that with like, there was just a bunch of adenines that washed over. then you can kind of like record, okay, first there was an adenine incorporated, then there was a guanine, and then that finds your sequence. So yeah, these are both like two faster ways to kind of do the same thing to sequence DNA. And that would be it for sequencing. So next we'll move on to blotting and labeling that's associated with blotting. So what blots do is they look for specific molecules like DNA, RNA, and proteins in a mixture by probing for them. So there's actually a bunch of different types of blots, but we're just going to talk about southern, northern, and western blots because those are kind of the ones that are mentioned the most. And these blots largely have like the same procedure. But what like their largest difference is kind of what they're looking for. So southern blots look for DNA, northern blots look for RNA, and western blots look for proteins. So a way you can remember this is this acronym. So I'm pretty sure this is a really popular MCAT acronym. So if you line up the word snow and drop, S matches with D, so southern and DNA. Northern matches, N matches with R, so that's northern RNA, and then W matches with P, so that's western protein. Yeah, so this is a pretty easy way to remember kind of, what blot does what. And so the process for each of these is first like electrophoresis after extracting, your molecules. And then what you do is you transfer kind of the results of your electrophoresis onto a membrane. And then you probe it with, different methods. And in steps 2 and 3, it's actually different for each type of blot what you do. For example, southern and northern blots use nylon as the membrane, and western blots use nitrocellulose or PVDF. And there's also various methods to like probe, but we'll talk about kind of how western blots are probed a little bit later. So I've included a little more on western blots because they are the most commonly talked about. So first, something that you do in western blots is after you stain your membrane. So in western blots, you have to stain your membrane after you transfer it. So you stain your membrane so you can see the proteins. And then what you do is you block the membrane with dry milk or bovine serum albumin to reduce background noise. And the reason this works is basically these are these are kind of like suspensions of proteins. So they bind with like basically everything that's not where the stain is, and then your proteins are. And then what you do in a western blot is you probe with antibodies. So we'll talk about that next. So immunohistochemistry is basically kind of probing with antibodies in tissues. And what you do here is you first label kind of your antigen on a tissue with a primary antibody. And this is the antibody made when like a cell host is exposed to a protein. So say I am exposed to protein X, and my body does not like that, so it makes antibodies that recognize a certain part of protein X, so it can be able to identify it and then get rid of it because my body does not like it. So, and then you label your primary antibody essentially with your secondary antibody. And so basically this secondary antibody is anti your first antibody. So if your primary antibody was an alligator antibody, then your secondary antibody is an anti-alligator antibody, and you're always going to see it like this. If the first one is a rabbit antibody, then the second one is an anti-rabbit. So The secondary antibody is like usually linked to like biotin or a reporter enzyme to allow it to be detected. And there's like, different ways it can be detected. Like here's fluorescence, here's like kind of just colors. And yeah, so the results you see kind of what the secondary antibody is labeled with. And then we have like kind of a really specific way to kind of do your labeling on these antibodies, and it's biotin streptavidin binding. So streptavidin is a molecule that binds to biotin, and what's good about it is that it doesn't really bind to anything else, like in tissues, which is good because you only want it to bind to biotin, basically. And then so you apply your first layer of primary antibodies, and then you apply your second layer of secondary antibodies. And these have biotins bound to them. And then you'll have a third layer that's streptavidin bound to something fluorescent, or streptavidin bound to an enzyme. And then this will bind to the biotins, and basically this allows you to visualize where the binding happens. So if it was an enzyme, you can add a substrate, and then the enzyme does something to the substrate that you can see. And that's it for immunohistochemistry and blots. So now we'll move on to how to look at protein production. So the first one we'll look at is ChIP-seq, and that stands for chromatin immunoprecipitation sequencing. So this determines what DNA sequences a transcription regulator regulates. So the process for this is basically you first cross-link proteins to DNA in living cells. So you kind of, keep those two bound really tight. And then you break the cells open, and then you kind of cut up the DNA. And then what you do is kind of, the immunoprecipitation part now. You use antibodies that are directed against the transcription regulator, and you use this to purify kind of like the DNA you cross-linked earlier. And then what you do is you sequence the DNA, and you can amplify this DNA with PCR. So then it would be easier for you to analyze. So next we have ribosome profiling. And what this does is it gives you an instantaneous map of ribosomes on each mRNA in the cell. And what it can also do is basically identify actively translated mRNAs in your cell. And so there's actually a few things that use of this test shows you. And it shows you that there's a lot of mRNAs in cells that are there, but they're not being translated until your cell gets a signal. And there's also a lot of open reading frames, ORFs, which are basically like links of DNA without a stop codon, that are too short to be counted as genes, but they actually make some small proteins. And it also shows that cells can rapidly and globally change translation patterns in response to sudden changes. So the process to this is pretty easy. So what happens is you add a bunch of RNA aces that are really good at breaking down RNA. So basically you break down, all the RNA in a cell, except for the RNA that's being translated by ribosomes, because that's being protected. And then these protected RNAs are released, and then they're converted to DNA, so they're more stable. And then the sequences are determined. So you can kind of see, here's, the mRNA sequences that were being translated before, the cell was, the cell's RNAs were, degraded. All right, so next is molecular interactions. So these tests will basically look at, how close are these two molecules, are they even interacting, stuff like that. So the first one is the Yeast Two-Hybrid test. So what this does is it identifies protein-protein interactions, and it can't do this in actual functioning cells in an organism just out in the out in nature, and you'll kind of see why, because you have to do, a lot of edits. So basically what you need to know about this one is that yeast have a gene for digesting galactose, which is it's kind of sugar, and this is controlled by the transcription factor gal4. And then this transcription factor it has an activation domain and a DNA binding domain. So basically it has two pieces. So it's this piece and this piece right here. And what they do is they basically, come together, and then there's like the activation domain kind of gets the signal to, come over to the DNA, and the DNA binding domain actually binds to the DNA. So basically what you do is you kind of take advantage of that. So you have what's called a bait and a fish plasmid. And then so a bait basically is your activation domain, so here, plus your bait protein, which is shown in green in this picture. And then your fish is the DNA binding domain plus a fish protein. And the reason it's a bait and fish plasmid is because, like, when you basically genetically edit a plasmid, so you insert this like protein after the DNA binding domain, and you insert this like bait one next to the activation domain. So yeah, you basically have these two, and they're attached to each other now. So if there's an interaction between these two proteins, then they're gonna bind, and then the DNA binding domain and the activation domain basically become part of the same complex again. So the gene gets to work. And the way you can kind of see whether there's an interaction is you like monitor the expression of the gene. So this gene is then called a reported gene, right? Because it reports, is it working? Like, are these two interacting, right? So a really popular way to do this is using histidine, and then because it's like a really important nutrient, and then the growth of the yeast colonies kind of like determines the expression of the genes, and that in turn gives you kind of like the lack or presence of protein interaction. Okay, so next we're gonna look at FRET, which stands for, I think there's two things it stands for: Forster resonant energy transfer, or like fluorescent energy, resonant energy transfer, I think. So basically what this does is it identifies interactions of molecules kind of similar to, the Yeast Two-Hybrid. And what you do is you basically tag two proteins or molecules with fluorophores, which are like, they're basically things that emit fluorescence, and they have to be different. And there's like kind of like a specific consideration you have to make, is that basically the emission wavelength of one overlaps the excitation wavelength of another. So on a graph it looks kind of like this. So if you look at this, so basically the wavelength that makes one of the fluorophores light up, it has to be the wavelength that activates the other one for this to work. So on this graph, it's going to be this like little overlap part between, the donor emission and the acceptor excitation. And basically if FRET happens, you will see light from the second fluorophore, because basically the way it works is, say this one's attached to protein X and this one's attached to protein Y. If they're interacting, then the definition of that is that they're less than 10 nanometers apart. And then that's enough space for that's close enough for basically, the light emitted from one of them to go into the other one and activate that one. So you'll be seeing, in this case, this yellow light. So if we're analyzing like a graph or something, just know that high FRET efficiency means more light from like the acceptor, which kind of entails like a closer interaction between these two molecules. Okay, so here's the last section, which is kind of like the miscellaneous technology section. So the first one is electrophysiology, specifically like the patch clamp technique. And what this does is it finds like a current or a charge across a membrane, and it's usually used with like, excitable cells, think neurons or cardiomyocytes and cells like that. So essentially the setup is you've got a pipette, and that pipette has like an electrolyte solution. So it can transfer, electrons like really easily. So it detects a current very well. And it's connected to something that can measure, electrical charges. So there's like a few different techniques you can use. So the first one is like I combine these two, which is like the cell attached method and the loose patch method. So basically in these two, you're not breaking the membrane at all with your pipette. So you just kind of do like a little bit of suction, and then you get like a little bit of membrane in the middle. And what's good about this is that your cytoplasm is still normal and you haven't disturbed it really. But what's not good is that you can't really control the intracellular environment of the cell. And then the second technique is you can use whole cell recording. So basically you suck it in really hard, and then you make it so the cytoplasm is continuous with the interior of your pipette. And then the next technique you can do is like the inside out recording method, where you basically, what you do is you kind of like put your pipette on the membrane and you like suck it pretty hard, and then it kind of breaks off this little piece. And then what you can do now is you can do things with like the inside of the membrane, since you see like this place is exposed. And then the outside out method is kind of like, the opposite. So what you do is you basically kind of do what you did with the whole cell recording. You suck up like a little piece, but like you break it off, and then you wait until these two parts like join again. So now you have access to, the outside of the cell while controlling, the inside. So you can do, things with the outside now. So next we have flow cytometry. And the goal of this is to sort and analyze cells by how they scatter light. So the setup for this is a little bit complicated. So first, you have like this thing called a fluidic system. And basically it sends like a stream of cells down in a line. And then you have like a laser that like points into the stream. And then you have like kind of like a detector, a scatter detector, like right in front of it. But you also have like a filter here. So, usually if there was nothing passing by here, the laser would just hit the filter. But then if there's something passing by, then, the light would scatter, and it wouldn't just scatter, it would, some of it would scatter and hit this detector, but it would also hit like, the side. So there's a, side scatter detector here. And then basically these detectors kind of take the signal and translate it, and they take it to a computer. And then you get kind of graphs like this. And what you can do is actually identify like clumps with different, properties. And these are like usually different types of cells. So yeah, you can identify kind of like cells, and like you can sort them down here based on how they scatter light. So this is a pretty good way to, sort cells and like, be like, oh, this is this cell, and, for purposes like that. Okay, so now let's move on to transformation and transfection. So the purpose of this is to insert DNA into organisms. And kind of like the first experiment you'll see is the Avery, MacLeod, and McCarty experiment. And I have a little vocab word here. Competence is when, like, the state when DNA can be taken up. So there's like a couple of methods that are used kind of to, carry out this. So the first one, I just have it listed under methods because you can use it with like any type of cell. So electroporation is basically you shock the cell with electricity, like here, and then you create like holes that are closed by cell membrane repair. So before they're closed, you can introduce things. into them. So, at the end you'll have a whole cell, but it has, what you wanted to be introduced inside now. And then, so a specific bacteria technique is using CaCl2 and a heat shock. So the reason you do this is because number one, DNA is negative and bacterial surfaces are too, and when things have the same charge, they like to repel each other. So CaCl2 basically it helps give the DNA like a more positive charge and just sends it in better. And then CaCl2 and the cold, so you would put your bacteria in a cold place before you heat shock it. It weakens the cell membrane, and then like the heat shock creates like a thermal imbalance that kind of forces the DNA to go into the cell. So for yeast, what you can do is you can add enzymes to degrade the cell walls, and that makes stuff called spiroplasts. And they're delicate, but they're good for DNA entering. And you expose it to like alkali cations, which again are positively charged, and that helps the cells take up DNA. So for plants, there's something you can do. You can use a bacterium called agrobacterium, and basically it kind of injects like its own DNA into plants, and it like changes the plant's DNA. So you can just like edit the DNA of agrobacterium to have a gene you want, and then you can use it to insert it into plants. You can also use like biolistics, which is basically kind of like shooting stuff into cells. So you use like a gene gun, which is like this basically. And you have like gold or tungsten particles coated with DNA in this case, and you shoot it into cells. And what's good about this is that you can transform plastids, so not just like the chromosomes, but what's not good is that it's less efficient than agrobacterium. So next for fungi, it's kind of difficult to introduce DNA into fungi, but what you can do is you can remove the cell walls, and it makes like protoplasts. So this is kind of similar to yeast. And then you can also use agrobacterium, but a big part of how agrobacterium works is that like plants, they produce like phenolic compounds after they're injured, but fungi don't do that. So you have to add those in if you're using agrobacterium with fungi. And you can also use CRISPR, so you can edit the genome. You can also use biolistics, and you can also use sonoporation, which is basically you make like pores in the cells with sound, I think. And then lastly, for animals, it's not called transformation, but it's called transfection, because for animal cells, transformation has like a different meaning. It means basically when like a normal cell becomes a cancer cell. So what you can do is use chemical reagents or cationic lipids, and the point of these is basically to give a positive charge. And then you can also use like physical methods like direct microinjection, and also biolistics, so you can shoot the DNA into a cell. All right, and then last one, the complementation test. I think this one's more genetics based than the other techniques, but basically the purpose is to see if the same like mutant phenotype is caused by mutations in the same or different gene. So what you need to do here is you need to mate two organisms with the same like mutant or different phenotype. So if you have like butterflies that usually have blue wings, and then you find two that have red wings, then you can mate those two to see if it's the same mutation or a different mutation, like in mutations in different genes that causes the butterflies to look different. So basically the way you analyze the results is that if you see that the animal has the same phenotype like as the parents, so the same mutant phenotype, then you can say that the genes don't complement each other, and they're in the same gene, like the mutations. And if the offspring has a normal phenotype, like the two red butterflies have offspring that are blue, then you would say basically there is complementation, and the mutations are in different genes. So if you look at this picture, you can kind of see like, a visual depiction of how this works. And that's it. I hope this video was helpful. Please leave a like if it was. Be sure to hit the subscribe button too if you want more competitive biology lessons. I wish you good luck on your competitive journey.